Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Jan van Houwerman from Ovisio.
Hello, good morning and good afternoon for everyone. Thanks for joining the webinar. So today we will be talking about the automation in monitoring of CAR T cells. For people who do not know who or what Ovisio exactly is and does, so Ovisio is a Belgian company reaching its 10th university at the end of this year. We are very much focused on our own R&D, so more than 50% of staff is still in R&D, hardware and software. A lot of the applications are built in-house. The main focus or our main market is in bioprocessing, where we have top applications in cell and gene therapy and in recombinant proteins, where the focus is mainly vaccines. We are starting up a small affiliate in the US, which should be completely started in 2020. In Europe, we are working together with a couple of partners for distribution. On the right-hand side of the screen, you can see the management. Today, we will be talking about two of our products that are on the market and that are specifically built for CAR T, with the Inline app and the QMOD. Both of these products use OS1 software built in-house that is combined with the machine learning platform. Here you can see in a quick infographic where our products actually can improve your process. The main focus of today will be the Inline F, which is capable of the inline closed-loop monitoring of your T-cell culture during the expansion step and as well during transduction and activation. The QMOD is more used in earlier or later stages. The Inline F, as you can see on the website, or as some of you might know already or have seen already, it's currently undergoing a very big update. We are moving towards complete cGMP compliancy. You will see in the image the alpha version that is already a working version and being used in a couple of labs. And in the middle, you see an image of the final design, which has been concluded and will be on the market in 2020, early 2020. The idea of the Inline F still is the same as with the current version, and that is that it enables you to take 1,200 images a day of your cells and of your culture, and it uses machine learning algorithms for a robust and reliable cell counting and phenotyping analysis of the cell. It can as well be connected to a wide range of bioreactors thanks to our single-use probe, and the software is CFR 21 Part 11 compliant. The reason why we do that is that we believe that to have a high quality product, you need to be sure that your starting material of the process is of high quality as well. And that's what will be possible by using the Inline F. So the Inline F actually comes with the BioConnect. The BioConnect is a single-use probe that takes care of the connection of your cell culture vessel with the microscope, and it actually guides the cells through a closed serial loop to the microscope where the images are taken, and then it goes back into the bag or into the cell culture vessel. So it's a single-use probe, single-use device, gamma sterilized in a closed-loop system that ensures a continuous flow, and in the end, there is no loss of sample volume. So all cells and all samples that are going to the microscope go back into the bag. So the microscope itself and the BioConnect, the development is reaching the phase that we will comply with all cell therapy requirements when it comes to cGMP. So we have a reduced footprint with an embedded PC. The Inline F contains both a pump, a computer, a touch screen, and an interface for the BioConnect. It's also possible, thanks to OPC, to have fully automation with external systems. It's possible to connect to external systems. The software is CFR 21 Part 11 compliant. The main setup that we use to communicate with other platforms and with other software and or servers and SCADA systems is OPC. I won't go into much detail around this, so if you have questions on OPC, feel free to contact me afterwards. This is in a nutshell what we have and what is possible. Now, if we go to the technology itself, it all starts with images. And when we are talking about images, what we take or what we do are actually 3D holographic images. And 3D holographic images is basically a combination of three images. You see an intensity or bright field image. This image allows us to see and identify some features and characteristics of a cell, but of course not so many, as we are completely label free and we are not using any stains or reagents. We do not touch anything of the cell. The bright field image does not provide us with a lot of information. We get a lot more information from the phase contrast image and of course from the 3D holographic image. So the phase contrast image you see on the upper right corner of the screen, and then 3D holographic image you see on the bottom left. The 3D holographic image, that's where we get a lot of information related to the shape and the volume of the cell. On top of those three images, there is also a refocusing feature, which means that for us it is possible that the cells floating around in the flow cell when we are taking images, and even when they are a little bit above or below the focus plane, it is possible for us to put them post-acquisition back into focus, which means that when you see an image, basically all the cells in that image will be in focus, and when all cells in focus, that leads to high quality image, which in turn leads to a high quality analysis. And high quality analysis leads to robust and reliable results. Everything starts, of course, with the image, but what we do with the image is actually even more important. In the image, the algorithms that are built with a machine learning system rely on the analysis of single cells. So all the cells within an image are analyzed individually, and that is based on a combination of 70 characteristics on a single cell level. Those characteristics, there are some subcategories in that, so there are a lot of morphological characteristics that are being taken into account for the algorithms, like the diameter, the cell perimeter, the circularity, membrane integrity, and so on. But on top of that, we see we have access to a lot of optical phase texture and intensity texture characteristics of the single cell, which allows us to dig a lot deeper and to get a lot more information on a single cell besides just the cell count and the viability. I've discussed already, or I touched already a couple of times, the subject of machine learning. So for people who are unfamiliar with machine learning methodology and how you can create algorithms via machine learning, this is a small, easy to understand infographic on machine learning. So basically, when we design a new algorithm, and that can be, as we did in the beginning, as easy as algorithm for viability, or what we're trying to do now, as difficult as finding small, almost phenotypical differences between cells. It always starts with the creation of a learning set or a training set. A learning set or a training set basically is a pool of images where we are pretty sure that all the cells within those images belong to one population. And that population can be a population of viable cells, it can be a population of dying cells, it can be a population of infected or transduced cells, or it can be a population of uninfected or untransduced cells. When we have defined those populations, we let the machine learning algorithm know, the machine learning platform know, okay, so now you have images of cells in status A and cells in status B. Please find within the holographic fingerprint if you can see any differences. When that is the case, and when you are looking at 70 characteristics on the single cell level, it is the case quite often that you find differences between two populations. Then we start with the validation of that algorithm. And that happens with system training. And system training basically means that we start providing to the platform images where there is a mixture of the population and increasing difficulties. So from 90% status A to and 10% status B, all the way up to 50-50. And that's how the system and the algorithm get trained. And in the end, there is the prediction algorithm, or basically the algorithm that's provided to the market and to the customers to really be used in the laboratory. That is all in the background. What is it, what you see, what you get in the lab? In the lab, you see, of course, in the software, your cells, the images of your cells. So that's the big image in the middle. On the left side, you see all the individual data points during one run. So one process combines data points almost continuously, but in practice it means every 30 minutes the software will generate a new data point where you will get all your data of that specific data point. This can be viability, it can be the viable cell density, the total cell density, the percentage of activated cells, like in this example. And then on top of that, we automatically generate the graphs that will represent how the culture is moving over time. So in this case, the viability goes up and the percentage of activated cells goes up. And all the cells are by software marked by their status. So in here, in this example, the green dots represent the viable cells, the blue dots represent the activated cells, and a red dot will activate a dead or a dying cell. Of course, we have compared our technology to a lot of current reference methods. In first place for viability and cell count. So we have done a lot of experiments. Most of them were working with wave bags or rocking motion bags, 2-liter expansion bag with a 1-liter working volume. There were some slight variations around that, but in general it was the same principle, the same idea. All working with CAR T cells, where the cells were grown for a number of days. Depending on the process, it was somewhere between 3 and 7 days. The goal was, of course, to continuously monitor the CAR T cells in the bag and count and discriminate the viability with strong correlation of an offline reference method. Could be a device from Beckman Coulter, could be a device from Thermometec, could be basically any method that at that point was being used by one of our customers. And you see that the automated so the reference method mainly automated cell counters, making use either of Triton Blue or acridine orange plus DAPI staining. And we see that there is a very strong correlation between the VCD, TCD, and viability data performed by those methods, taking into account that those methods require manual sampling and having a sample moment only once, maximum twice a day instead of continuously. Showing a very strong correlation. What is probably even more interesting is that the Inline F is perfectly capable of accurately monitoring the difference in different processes. So if you have of course you have different donors when talking about autologous CAR T processes, but as well within or between companies or even within companies, you have different processes for different products. It's perfectly possible to monitor the differences between the processes, to keep track of differences, and to do that in a very reliable and robust way. In this case, the red dots are the sample points and the data points generated by the offline measurements, the reference data, and the blue lines represent the results generated by the Inline F and OS1. For the last couple of slides, I have some interesting topics where we go beyond the measurement of just viability and total cell count. As you can see here, it is possible to differentiate subpopulations within your CAR-T culture. In this particular case, you can see that it is perfectly possible to track small contaminants. In this case, it's beads that are being used by numerous of our customers to activate T cells, to keep track of the beads in culture, to count the beads, and to differentiate them from cell debris, to differentiate them from cells, and to mark them correctly. And that way you can keep track of the ratio beads against cells and see if the culture is really growing or not. Another point we are working on, and that is already on the market and being used, is a tracking of the activation status of T cells. So again, here the blue cells, the cells marked in blue, are cells that underwent an activation, and it's possible to just count the activated cells or count the amount of activated cells within a culture. So to conclude, I can only say that the Inline F meets the needs of cell and gene therapy manufacturing. So it's an inline system in real time. It's a closed system. It's automated, so it's a completely walk away. You start it when you do the inoculation. You come back when you're ready to go to downstream processing. It's completely label-free. We just use the images that we take with the microscope. No need to have the use of stains or reagents or fluorescent dyes. And thanks to the machine learning, it's possible to differentiate subpopulations within a CAR-T culture. Of course, that leads to increased automation, increased reproducibility, increased product characterization, which causes for a decrease in costs and a decrease in manual steps. Then I would like to mention some of our reference customers. Not all of them are in CAR-T. We are working on vaccines as well. If you have more questions, I'm available for the next 10 minutes, but please feel free to reach out or meet us at any of those events the next coming weeks. So thank you very much for your attention, and I'm happy to answer any of the questions you might have.
Thanks, Jan. So the first question is, does your 3D holographic analysis help determine the phase of the cell cycle?
We have done some experience on the phase of the cell cycle. It's not a finished algorithm yet, so there is still some work to do. But it's definitely not entirely impossible. I would say reach out to me to discuss some of those details because it will lead us too far on here.
And since you have no dyes, what imaging factors are used to determine viability?
Yes. So there are some very specific characteristics that distinguish a viable cell from a dead cell. One of the things, one of the most easy to explain, is that a viable cell, which is nice and round, will create a refractive peak when it's hit by light, while a dead cell or a dying cell that's losing the circularity and membrane integrity will diffract light when light hits the cell. And the difference between the refractive peak and the diffraction of a dying cell can be assessed by by the algorithm, and that's how we can differentiate between a dead and a viable cell.
Okay, and it looks like we just have one question. If you haven't typed in your question, you can type them in now. If we don't get to them on the talk, Jan will be able to follow up with you directly. It looks like we have a couple more questions, but I think this might be the last question we have time for, so we'll see. If I understood correctly, the sample flows through and then goes back to the culture vessel bioreactor. How is sterility maintained?
Yes. So the probe, which is in contact with the cells and with the culture, is completely sterile. So it's gamma sterilized. It's completely sterile. You connect it to a bag in a sterile way. We have multiple options, either welding or louver locks. There are a lot of possibilities to think of. But once it's hooked, you do not touch it anymore, and the cells do not leave the probe or the bag. So there is no contact between the cells and the outside world, and that's how we keep the sterility in place.
Okay. If microbial contamination occurs, does the system detect it?
The short answer is no. The longer answer is if it has really an effect on the cells itself, then we might pick it up. So cells will start dying, and we might pick it up a little bit earlier. But the magnification used by the microscope is not capable of detecting or seeing a bacteria or microbial contamination or infection.
Okay. So these are great. I see a number of really great questions coming in. I think we do just have time for one or two more. So keep typing them in, and again, we'll pass this to Jean. Yeah. Okay, so would the technology be able to distinguish a virus-infected cell?
Yes. When using, and then we are talking about gene therapy and vaccine production, so when using viral vectors, one of the things that we are able to detect, and we have more information on the website for that, and a very nice use case. So SF9 and baculovirus, there we can really see a difference between an infected and a non-infected cell. The same goes, but those algorithms are still in development, goes for HEK cells in combination with lentivirus or AAV.
Okay. And this is the final question we'll be asking. One of the key challenges in the cell therapy industry is scaling up production so many people can access the therapy. I understand that Inline F can only monitor one bioreactor at once. Are there any plans to facilitate higher scales, either monitoring more than one bioreactor at once or a next generation product that can monitor tens of bioreactors at once?
As of now, it's indeed one-on-one. There are no plans to multiplex. So we reduced already the footprint quite drastically, and we see that is already a big solution and a big step forward. Multiplexing our device creates a whole bunch of other challenges. I won't go into much detail at this right now, but as we are working with very fine optics and very fine optical setups, as soon as you start to multiplex, you need those optics to be able to move around, which combines with very high cost. And in the end, the cost of a multiplex device is much higher than the cost of multiple one-on-one devices.
Okay, well, thank you very much, Jan.
You're welcome. It was a pleasure.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International SC Expert webcasts. Look for those announcements in your inbox. Goodbye.